Okay, so since it is already 3 o'clock, let's get started right away with the TA session. I'm seeing no one in the meeting hall right now, but anyways, I will proceed with the TA session. So, one second, I'll just yes. So, good evening, everyone. So, I am Arjun. I am the one of the TAs for this course, Bioengineering, an interface with biology and medicine. And welcome to the week 3 TA session for this course from 3 p.m. to 5 p.m. So, as usual, as we went for the past 2 weeks for the two TA sessions, the routine will be pretty simple. So, what we will be doing in this TA session is I'll be going over a brief recap of the contents that were taught for week 2, week 3, and after that we will be looking at some example problems from the previous year's assignment of week 3, following which we will be discussing the and com- compared to the week 1 and week 2 TA sessions, there will be a small addition to this week where I'll be also discussing the week 1 answers for this year's iteration of this course assignment. So that from this week onwards, we'll be discussing the subsequent weeks' assignment sessions as well. So in between there will be some doubt sessions here and there. So whenever you have doubts, you can feel free to open your mic or put your doubts in the chat box, and I'll be ready to clarify them whenever I see the doubts. So without further ado, we'll just get right into it. So week three's content was uh, sequel from a kind of contents of week 2, where we kind of learned about some basic concepts in week 2 about polymerase chain reaction, and this was sort of a continuation from week 2 where we saw the potential applications that that the techniques such as polymerase chain reactions could do. The course instructor Dr. Sanjeeva Srivastava started from polymerase chain reaction, and he went on to explain about the importance of primers and how the design of primers and how the uh, changes that are incorporated into the primers while designing them helps us to study the effect of those the changes in primers. So you have a normal primer and you have another primer which has some changes in it. So if you incorporate changes in particular bases, you will be able to see the effect of those changes of those bases between the normal and the changed primer. And how do you do that? You continue doing a regular PCR, polymerase chain reaction, and based on the amount of quantifiable DNA that you get, you are able to observe changes in the final protein that is able to be synthesized from your nucleotide sequence. So, following the importance of PCR and primer design, the course instructor went on to discuss about several fluorescence tags that are used in the process of polymerase chain reaction as well as in DNA cloning. One of the notable tags that were used to explain processes are the 6 histidine tags, and he also explained how a particular technique known as affinity chromatography, where we use metal ions such as nickel, which is used to bind to the 6 histidine tags, is used to elute the desired DNA sequence of our interest, which is being bound by this 6 histidine tags. And after that, we also saw potential applications of this recombinant DNA technology, where we saw the essential proteins that are being used by a lot of patients worldwide and on a day-to-day basis. One of them, which was insulin, and we also saw another protein called this BCR-ABL protein, which was and which had an inhibitor called imatinib, which prevented the phosphorylation of this BCR-ABL enzyme, and how imatinib is also being produced on a large scale. Following that, we also saw how uh, transgenic animal technology, for example, in this case, transgenic goats, were used to produce an alternate form of goat milk, which had specific proteins of our interest. In this case, they had the goat milk had antithrombin production, which was used to produce this antithrombin in bulk amounts through transgenic goats. So, following the applications of DNA cloning as well as PCR, we went on to see advanced techniques in sequencing as well as finding the sequence of a particular DNA sequence. So, the first among that, the course instructor talked about the Human Genome Project, in which we talked about how there was a widespread interest among all the scientists to know about the complete DNA sequence of any human being, and how as time progressed and as technology progressed, as well as the cost of sequencing reduced, the goal shifted from knowing about the entire genome of a human being to comparing genomes between two different individuals to study the changes in the genome and how it leads to the formation of specific conditions or diseases, etc. and etc. And following that, we saw how specific we saw and we went into different sequencing techniques, such as one of them, which was this Sanger sequencing techniques. This was a pretty useful. This was a pretty famous technique and a very elaborate technique as well. The schematic of which is given here. So what we basically do in Sanger sequencing is we denature the double-stranded DNA using heat, and following which we generate multiple copies of segment. How do we do that? And we saw it last week using polymerase chain reaction. So what do we do after that is we have some we initially use primers, but what do we do here is in addition to the DNTPs, like we saw in the last week's TA session, there was this one question about DNTPs and DDNTPs. So in Sanger sequencing, what we do is in addition to DNTPs, the deoxyribonucleotide triphosphates, we also have something which is called as a dideoxyribonucleotide triphosphates, DDNTPs. So what happens in the DDNTPs is the both the oxygen atoms in the ribose sugar base is gone. So the incorporation of DDNTPs into this chain into this chain generation during the extension phase causes the termination of the chain sequencing. So as we add the polymerase solutions, and as we add the different DD and TPs, what happens is after each step, multiple copies of DNA are generated in which different stages of the chain termination is achieved. So what do we do is we again denature this target DNA and we run them in a agarose electrophoresis. So based on their charge to molecular weight ratio, they run, and since these DNA chains are the complementary chains, we are able to find based on the molecular weight of this uh, uh, based on the molecular weight of this grown chains, we are able to elucidate the complementary sequence of the desired DNA sequence that we want. So this is how Sanger sequencing works in general. And after that, we also saw examples of how next generation sequencing happens. So in next generation sequencing are divided into several generations, but common thing is we saw how all of them have common steps such as library generation, amplification of your DNA using PCR, as well as detection using uh, small quantities of your target DNA. And moving on, we also saw further applications of this gene sequencing, especially to study the gene expression. And we saw different types of techniques. We saw qualitative techniques, quantitative techniques, and one of the qualitative techniques that we saw was called in situ hybridization. So if we have a slice of a desired region where we want to visualize what kind of DNA is present where, we use labeled DNA parts and we use them to coat or bind. We use antibody or DNA to bind to the desired region of our interest, and based on how these regions light up in under a microscope, we see the presence or absence of our desired DNA sequence. And when coming into the quantitative techniques, we saw something which is called as a reverse transcriptase PCR, where we have initially we have a mRNA of our interest, and we use something called as a reverse transcriptase, an enzyme which converts the mRNA into a desired, which helps in the production of our complementary DNA, that is cDNA, based on the mRNA that we have. We also saw how specific other components such as the oligo DT primer, where in addition to the primer that we already have, we also have a uh, oligo DT, which is a deoxythymidine, which is used to bind to the polyadenyl train present in the mRNA sequence. And after looking at reverse transcriptase PCR, we also looked into DNA microarrays, where we can image the presence of uh, DNA sequences even if we have very small amount of DNA present in them. So that's all the and after discussing all these techniques, we also went into discussing the potential applications of this recombinant DNA technology, especially in the field of especially in the field of genetically modified organisms and genetically modified crops, which are used to negate certain negative traits such as environment resistance and develop resistance towards environmental stress. We saw in detail about how agrobacterium diffusions helps in giving resistance to pests, and we also saw the case of golden rice. And finally, we ended with a clinician's perspective of how they had a startup and how they work, and how they what are their main goals in that startup. So that's all the uh, that's all is there about the recap of week three of the lectures. So I'll leave a short uh, doubt session till now. The time is 3:12, so I'll leave a short doubt session till 3:20, following which we will go over some problem statements from week three assignment sessions. So I'll leave the room. I'll not leave the room. I'll be here. But if you have doubts, feel free to either open the mic and ask, or type your doubts in the chat box.
Hello, sir. Am I audible, sir?
Yes, 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 you're audible.
Sir, I had a question, sir. This is Orko, sir. I
Yeah,
am
yeah,
a
yeah.
second year genetics student,
Okay, okay, yeah.
bachelor's genetics student.
Yeah.
So my question is, we all know that we use endonucleases to sort of excise the DNA bonds. I mean the DNA strands, right?
Yeah.
Why can't we use topoisomerases in case of this endonucleases to cut the bonds?
So, everything is based on this um, your recognition sequences. So, topoisomerase also have a sort of different functions. Ba- like compared to your normal enzymes, now compared to your normal what to say, compared to normal and restriction enzymes, your topoisomerase have a different kind of function, right? So apart from making um, making like excising things, um, most of the topoisomerases will also be cutting your single strands. Like um, I think they are divided into like class one and class two, and out of
Yeah,
them, like most
yeah,
of them are class one, and
yeah.
they make only like single stranded cuts, right? But in
Sir,
the
but there are DNA two. I mean DNA topoisomerase two enzymes like DNA gyrase as well, which can cut both the strands at once. So why can't we implement them into RT-PCR or something?
So ultimately, what like a simple answer that I could say is uh, everything depends on the site of excision. So if your topoisomerase can recognize a sequence which can cut your which can cut into the plasmid, then that can also be used. But most likely that will not be the case.
Okay, thank you, sir.
Yeah. Actually, I also forgot, like, I hope that I was audible actually all the time while I was explaining the recap of the lectures. Any other doubts? So we have 2 more minutes till we can go and discuss the answer problem questions. Actually, with the Orco's question, was there was there doubt about why topoisomerase is not used in place of restriction enzymes, or like if Orko is still in the call, can hear me. Like, um, was the doubt actually about topoisomeresis not being used instead of restriction in nucleases, or tos the use of topoisomeresis in general in PCR?
No, sir. For example, I've always heard about endocyl nucleases being used everywhere,
Yeah.
but recently I got to know that topoisomeresis can also, cut the cut the DNA strand. So I was wondering why topoisomeresis are not used in the general terms. So yeah.
Okay, okay. I mean, then I think the sequence specificity will only be the only answer. Um, topoisomeresis might not be the way to go because of the sequence specificity. In very very rare cases, if the sequence is specific enough, then topoisomerase can also be used. Class 2 topoisomerase can also be used in plasmids. Okay, so 320. Anyways, if we have any other doubts or discussion furthermore, we can also like discuss it later in the later parts of the TA session. So we'll just go into the problem statements of week 3. So basically, this is just a um, the assignment questions that that the previous iteration of this course had. So we'll just go into it. So question number one. So Ramesh planted a hybrid tomato plant in his garden. He wanted to perform the sequencing of the plant. As a result, he performed sequencing of the DNA extracted from the plant. He set up the mixture by taking DNA polymerase enzyme, a primer, 4 DNA nucleotides, template DNA, and DDNTPs. After successful reaction of the 4 tubes, he ran an agarose gel to get the results. So everything is just um, properly like a proper replication of the Sanger sequencing method that we saw in the previous slides as well. So he has taken out the sample, he has used the polymerase enzymes, primers, the all the DNTPs, GDNTPs, template DNA, and after the gel reaction is done, be done, he has also run an agarose gel. So they are asking us to look at this. This is the final gel that he has got. So from this, they are asking us to interpret the sequence of this DNA. So what could be the possible DNA sequence? So does anyone want to go around saying how to go this question? I'll give you guys some time to just look at the gel. So, couple of points that while we are thinking how to solve this problem, a couple of points that I like to reiterate while solving any problem related to Sanger sequencing is you always need to. So, agrosgel electrophoresis always separates the samples based on their molecular weights. So the farther your band travels, the lower molecular weight it has. Therefore, it is uh, more towards the 3' end. Like your and the second point is whatever band you are getting here, that point in the sequence will actually be the complement of the original sequence that you are searching for. So those are the 2 points that you need to keep in mind while finding answers for something related to Sanger sequencing. Like I'll give you guys, now it's 3:23, so let's just say 3:27 we'll start solving the problem. You guys can figure out how to like do it, keeping these 2 points in mind. So further the molecular weight is more 3 prime in nature, and once you kind of get how to look at the problem, it's kind of simplistic only. Like
Sir, I think you can go ahead with the explanation, sir.
okay, sure. So yeah, um, so 2 points. One is the farther you go, the more three prime it will be, and whatever band is coming here, so the complement you should consider the complement as original sequence. So the farthest here, the first band will be G, so the complement will be C. So and following that, you if you solve the entire sequence, it will be C T G A, and then G T C sorry, yeah, C A, and then G T. So this sequence kind of matches with the answer. So this will be your correct answer for the this question. So I hope you get how this Sanger sequencing works now. So whatever bands are coming in this picture, the you should consider the complement of it, and from the three prime end. We'll come to it later. We'll come to this question later if any of you other guys have doubts. So I'll move on to the next question. So read the given statements carefully. So they are given five statements. Statement one is NGS uses chain termination method for sequencing DNA. Statement 2 is NGS platforms generally sequences by the principle of sequencing by synthesis. Statement 3 is Sanger sequencing method is also known as chain termination method. Statement 4 is NGS sequencing platforms make use of fluorescence resonance energy transfer for sequencing. And statement 5 is recent technologies like third generation sequencing technology makes use of protein nanopores for sequencing. So, question is actually how many of the given statements are incorrect, not correct. So, this is also a pretty straightforward question. So, if we just keep looking at the statements one by one, so NGS uses chain termination method for sequencing DNA. So, this is actually incorrect. So, chain termination method is actually used by Sanger sequencing method, which doesn't come under this NGS sequence. So statement 1 will be incorrect, and since like we can also correlate with statement 3 saying Sanger sequencing method is also known as chain termination method, which will be your uh, correct statement. So statement 2 is NGS platforms generally sequences by the principle of sequencing by synthesis. Yes, it is correct because you have to synthesize a library first before going on to sequencing. Statement 4 is NGS sequencing platforms make use of uh, fluorescence resonance energy transfer for sequencing. This is actually incorrect statement because for sequencing we don't use FRET for NGS. Statement 5 is the recent technologies like third generation sequencing makes use of protein nanopores for sequencing. This we already saw in the lectures as well, where the course instructor said. So this is the correct answer. So the incorrect statements here are statement 1 and statement 4. So the correct option here, that is two incorrect statements. So if none of you have any doubts with this, I'll move on to the next question. So consider a given stretch of mRNA sequence. So they given a mRNA sequence here, and which of the following shall you consider a single stranded DNA probe for the given mRNA sequence? So this is also a pretty straightforward question. If you have already solved the Sanger sequencing question, you could like go go around solving this very easily. You just have to use the rule of complementarity, and uh, one more thing is yeah, you have to use the rule of, one thing is you have to use the rule of complementarity. So it's 3 prime to 5 prime, you have to use 5 prime to 3 prime, and yeah, if you guys look at the answer, there are only like uh, 3 prime to 5 prime, and uh, one thing is from mRNA, if you are considering a probe for DNA, you have to negate, you have to consider the possibility of uracil. So uracil will be thymine when it comes to DNA. The complement of uracil will be thymine, will be when it comes to this thing. Not
Sir,
the
I think
complement.
second one is the answer, sir.
Uh, but how do you say it's actually there's a kind of a small twist in this question, or go like so they think once again before this thing. So I'll explain the question as well. So when you, I have told you that you have to consider the rule of complementarity. So the whatever answer, whatever DNA sequence you are reading, you should be able to read it from the 5 prime to 3 prime. They have given sequences which are from 3 prime to 5 prime as well, but if you read it from 5 prime to 3 prime, and if you can match it with this sequence complementarily, then that is also kind of fine only. So if you say from a, your DNA will have a 5 prime 3 prime end, and at the edge of the 5 prime you will have some, your complementary to uracil will be adenine. So your 5 prime adenine is what will be your uh, what will come in your correct answer. Your 5 prime thymine will not come. And as we just move on to the end of the sequence, your complement to this will be the 3 prime end, and the complement to adenine will be your thymine. So where your thymine is, and automatically you can negate these two options because these are RNA sequences. This is not a DNA sequence, but the answer asks a single stranded DNA probe. So your answer will actually be option A, because if you read it from the 5 prime to 3 prime end, this will also be complementary to the original mRNA sequence. So I hope you got how the question works.
Yes, sir.
Yeah. So I'll move on to the next question if there are no further doubts. So You visited a research lab at IIT Bombay. As a curiosity, you collected two different species from Powai Lake and Arabian Sea. Now you have extracted the DNA out from two species. Now, if you choose the DNA hybridization process to identify its relatedness, which of the following statement is correct? So they have given four options, like two DNA species from two different this thing, and they are given four options. One, it will form more hydrogen bonds if the two species are closely related to each other. It will form less hydrogen bonds if two species are closely related to each other. Not at the DNA level, but mRNA hybridization is the way to understand the relatedness of the species. DNA hybridization technique cannot be used to identify the related species. So, which one of them will be correct? So, the actually for this question, you might tend to think a lot, but it's a very simple question. So, if you go around the answer, like basically you have two DNA species. So, if you just denature them and manage to bind both single strands from different species, and if they bind to each other more closely, it's going to be it's going to tell you that they are more closely related to each other. So the correct option will be the option one. It will form more hydrogen bonds if the two species are closely related to each other. So if you take this DNA species and if you bind it with the complement of this DNA species, then both of them should bind to each other more and form more hydrogen bonds if they are closely related to each other. Related to each other. So I'll move on to the next question. So this is also a pretty straightforward question. You just have to identify the uh, mistake in this question. So Rohan and Mohan are two research scholars in IIT Bombay. They have set up two reactions for cDNA synthesis. The steps followed by Rohan is they have mentioned addition of RNAs, reverse transcriptase, oligo DTs after synthesis of first DNA strand, removal of RNA, and a reaction is continued. Mohan has done the same thing, but instead of oligo DTs, he has added oligo DCs. And but the suggestion of their colleague Sriparna, Rohan made some modifications to the reaction mixture and made the following modifications. So the modified steps followed by Rohan is uh, addition of mRNA and reverse transcriptase. Same oligo DT did not remove the RNA after the first synthesis of first DNA strand and let the reaction continue. So out of this, we should choose the correct statement, like whose synthesis would not have had worked, and like the correct cDNA synthesis, either Rohan's or Mohan's, and which step of them would not have led to proper cDNA synthesis. Like this is also pretty straightforward. So you already know that for cDNA synthesis, we usually use we saw in the recap as well. So we use something called as a oligo DT primer, which is used to that oligo DT is a set of it's a train of thymidine sequences which is used to bind to the poly A tail, polyadenine tail, which is a characteristic of mRNA sequence. So in Mohan's case, instead of oligo DT, he has added oligo DC. So um, the reaction wouldn't have had continued because the primer wouldn't have bound to the sequence. So the second option is the step, uh, step 2 followed by Mohan would lead to the, step 2 followed by Mohan would lead to the incorrect synthesis of cDNA. So option 3 is the correct answer here. So if there are no doubts with this question, Basically, instead of you just have to identify the mistake in this question, and then that will lead you to the answer for this one. So I'll move on to the next question. So Vijay wanted to check the gene expression similarities or differences between a male and a female monkey. As a result, he probed the sample from male monkey with Psi 3 and female monkey with Psi 5. So Psi 3 is a green dye and a Psi 5 is a your red dye, and he expected three colors in a microarray plate. Greater gene expression in male monkey exhibiting green color, greater gene expression in female monkey exhibiting red color, and no gene expression exhibiting gray color. But after scanning, he was astonished to see another color emerging, that is yellow color, emerging on the microarray slide. So, what could be the possible reason for this? So this is also a pretty straightforward question. It is also like this concept was also discussed in one of the lectures as well. So options are the Psi 3 and Psi 5 dyes reacted with each other and formed artifacts. Uh, there was some contamination in the samples that deposited on the spots exhibiting yellow color. It is protein that is showing up the light color on the spots. The gene showing similar expression in both the male and the female monkey exhibited yellow color.
So
What
the
could be
fourth,
the yeah, exactly. So, if you have genes expressing from both the male and the female monkeys, and both of them are from a similar thing, they would express equal amounts of red as well as green color. That would lead to a mixture of signals. So it will that is what is causing the expression of the yellow color. So the option is option four. Uh, Similar expression in both the male and female monkeys mixed together and form the yellow color. So I'll move on to the next question. Swati has extracted Swati has extracted RNA from Rattus novergicus. She wants to quantify it for further downstream process. From prior experience, she knows that not more than 100 microgram of RNA can be yielded from a fully grown rat. There is a problem in the quantification machine they are using, and its absorbance reading is not showing beyond zero point 0.5. She intends to dilute the RNA sample in 100 microliter of distilled water. What is the ratio in which she should dilute the sample to get the absorbance range within zero point 0.5? So I mean, this is also a kind of like there is no like hard thinking and all. So if you want to get lesser and less absorbance, you have to dilute it more and more. So the highest dilution factor is what you will choose in this question. So the answer is 1:50. Like there is no hard uh, if you can perform hard calculations and all, but there is no need for that. You just go with the highest dilution for this question. So if there are no doubts, I'll go on to the next question. So this is also a very pretty straightforward question. Which of the following makes Agrobacterium tumefaciens a useful resource for transformation? So TA plasmid cry-gene, tDNA, and chemoreceptors. So this is a pretty straightforward question. TA plasmid is what your gives your Agrobacterium tumefaciens a useful resource. So you um, move your desired DNA of interest into this TA plasmid, and then you proceed with cloning to generate this induce tumor in plants, which is going to cause this production of nanocrystals which will kill the pests. So I move on to the next question. One second. Okay. There is a bug in my that the I don't know what happened. There is a bug in my PowerPoint presentation. It's okay. I'll just reopen it and present it again. I'll just present it quickly. Yeah. So sorry for the quick this thing. So I'll just yeah. So question number 9 is read the following statements carefully. So there is a assertion and a reason. The assertion is Bt toxin, that is a toxin produced by Bacillus thuringiensis, produced in plants as a harmless prototoxin, but becomes toxic in the insect gut. The reason is the protoxin is converted to a toxin in alkaline conditions, and the insect's gut provides the appropriate conditions for it. So, from the assertion and the reason, we have to choose the correct statement. And this is also a pretty, straightforward question that we saw from the lectures, and that is both of them are true, and what the assertion reason says is the correct explanation for the assertion. That is, the Bt bacteria is producing the prototoxin, and once it goes into the toxin, its insect's gut is acidic in condition, so that converts the prototoxin into active toxin. So it swells up the gut of the insect, so that causes the inflammation of the gut causes the death of the insect. So pretty straightforward. So I'll go on to the last question of the problem statements for this week. So Sheila, a plant biotechnology student, has been closely observing the cotton plants in her grandfather's farm. She observed that due to insect infestation, her grandfather loses 30 to 40% of the yield. She wants to develop a transgenic cotton plant to help her grandfather overcome this issue. Which of the following can she try? So they have kind of given like four options.
So the second one, sir.
Inserting the gene of interest using a bacterium into the contact plant genome. Yeah. So pretty straightforward once again. So uh, yeah, so that's all I have for the problem statement for this week. So once again, I'll give a short break till like 3:50. We can discuss the. Uh, I'll just go over the week one assignment answers for your iteration. This thing, the actual week one answers. Okay, I'll just quickly go over the questions of the last. Uh, so, yeah, high-speed bullet trains like the first one is high-speed bullet trains like the Shinkansen in Japan used to make sonic booms as they emerged from tunnels, which was problematic for the locals. To remedy this, the Shinkansen was redesigned with a pointed front like a kingfisher's beak. This innovation revolutionized the world's fastest bullet train. The correct statements. So here options A and C are correct. That is the sonic issue was addressed by a method called bioengineering, and the biological feature mimicked was the beak's design for its natural efficiency to slice through a water to catch a fish without making any noise. So question number two, what could be the possible advantages of engineers knowing biology? Choose the correct combinations. Growth in biorobotics, development of bioweapons, development of biofilters. So here, obviously, except for the bioweapons, that is creating the deadliest of viruses to deal with alien attacks, both the growth in biorobotics as well as the biofilters are advantages. So option 3, that is both 1 and 3 are advantages to mankind, are the correct options. So to check the question number 3, to check the packaged milk quality of a particular brand, you put a drop of milk diluted with sterile water onto a glass slide and stain it. It is then placed under a microscope at 40x magnification. What could be the best possible prediction for the microorganisms mounted on the slide based on its morphology?
So the fourth one, sir.
A mixture of gram-positive cocci and gram-negative bacteria. Yeah, definitely. So, fourth question, DNA sequence and structural comparisons categorize all living organisms into three primary domains, bacteria, archaea, and eukarya. And environmental studies show that the archaea are abundant and varied in soil, seas, and fresh water, in addition to harsh conditions. Choose the correct statement about archaea. So, option the correct option is organelles and other internal membrane-bound structures are essentially present in archaea cells. So, archaea are somewhat of a bridge between bacteria as well as eukaryotic organisms, but that didn't mean that they had the advanced organelle system that the uh, eukaryotic organisms possessed. Question number 5 is, you work in a laboratory where you are instructed to culture an unknown strain. After 48 hours of streaking in a culture plate, you observe that the bacteria has an extended thread-like morphology. Um, thread-like morphology is here. Which is this organelle called, and what are the what kind of functional or structural benefit does it impart to the organism? Obviously, we can see that these are like flagella. This thread-like things are flagella, so they are like used for movement kind of movement kind of function. So, correct option is it's flagella with rotatory movement function. The next question is, the endoplasmic reticulum is a substantial dynamic structure serving many roles to itself. Choose or point out the correct functional and structural information for this organelle. So, uh, for the endoplasmic reticulum, we have to point out a structural and functional this thing. So, already we know that endoplasmic reticulum is divided into like two different components. One is the rough one and the smooth one. The rough one will have this ribosomes on top of them with to produce the proteins for protein synthesis, and the smooth endoplasmic reticulum will be smooth and they will not possess ribosomes, and those are mostly used for lipid synthesis. So option 1 is the correct option, where structural basis is divided into two, rough ER with ribosomes and smooth ER without ribosomes. And the function is to synthesis, folding, modification, and transport of proteins. So, seventh question is, Choose the cell organelles that are majorly involved in intracellular transport. So already we know which are involved in intracellular transport, which is the endoplasmic reticulum, Golgi apparatus or Golgi apertures, and lysosomes. So question number 8. The primary function of the organelle flagellum is to facilitate mobility and chemotaxis. Name the element that is involved in creating the structure of bacterial flagella. So there's also it's a pretty straightforward question. Flagella is produced from a protein called flagellin. So question number 9. Which statements are correct regarding the origin of replication among prokaryotes and eukaryotes? So they have given four statements. Prokaryotes have a single origin of replication, like ORI. Sorry, origin of replication. All eukaryotes have a single origin of replication. All prokaryotes have multiple origins of replication, more than one, and eukaryotes have multiple origins of replication. So they are asking us to choose the correct statement. So out of this, like only one, like two statements are correct, which is that prokaryotes have a single origin of replication and eukaryotes have multiple origins of replication, which has led to eukaryotes having greater diversity than prokaryotes. The last question is the Human Proteome Project (HPP) is a global initiative led by the Human Proteome Organization that intends to change our understanding of the human proteome. What are the major missions of the project? Choose the correct statements from following options. So they are given four options here as well. So it's a collaborative effort including numerous research labs, which is kind of correct. And its goal is to systematically map the whole human proteome by utilizing existing and new approaches, which is also correct. This project has one primary aim to cover the information about the human epigenome, which was completed in April 2003. This is not a correct answer because it was started around April 2003, if I am not wrong. And 2003, like not started per se, but only significant progress was made around 2003. So this is a incorrect statement. Fourth option is it aims to discover the missing proteome of the human body and creates the groundwork for the development of diagnostic, prognostic, therapeutic, and preventative medicinal applications. So this is also a correct statement. So the correct option will be 1, 2, and 4, which will be your second option. So uh, I think this assignment answers you would have already received in your NPTEL portal since the deadline has already passed. But for some questions which uh, half of them were straightforward questions, and for the other questions which needed sort of explanation, we were able to explain it. So that's all I have for this week. But we have uh, room till 5 o'clock for those discussion. So I'll leave this room open till at least 4:30 or 4:40. Um, if none of you have any doubts, we can. I'll see you guys on the next Saturday, which is 15th February from 3 p.m. to 5 p.m. And I'll right now I'll leave this room open for uh, general doubts.
Sir, if you have the like question paper of the second week, sir, can we solve the third question from that week?
Actually, I was about to do that next week actually because I was the plan. My plan was to actually solve this. I can go now actually. If,
The only I am there in the room, so like
yeah, yeah, yeah.
no one else is there. So
Exactly. Yeah. So I don't have it in the PowerPoint format. So we'll just go over that question if possible. Oh yeah. So third question says that Ramesh has been provided with the map of a plasmid P3000 whose size is 15 kb. He observed the following fragments with different combinations as mentioned below. So NOT1 and plus EcoR1 gave 4 kb and 11 kb. BAM H1 plus STU1 gave 9 kb and 6 kb. And EcoR1 plus STU1 gave 6 kb and 9 kb. So upon cutting the plasmid with BAM H1 EcoR1, which of the following fragments would Ramesh observe? Okay. So okay. So yeah, so we have like for this question, how we solve this? Like I'll just try to generally draw a plasmid map. I hope the plasmid map is okay. I'll just draw it here. Yeah. So let's just say this is a general form of a plasmid map. So they are saying that eco R1 and eco R1 and STU1 gave 6 kb and 9 kb. So out of this 15, I'll just put this as yeah. So let this be eco R1 and let this be STU1. So if we cut eco R1 and STU1, it's going to give us two uh, two different bands. One with your 6 kb and one is 9 kb. So this will be 6 and this will be 9. And we'll go on with STU1 itself. So BAM H1 and STU1 give 9 kb and 6 kb. So uh, for change, we'll just say this is BAM H1. For simplification reasons, we'll just give it as BAM H1. So BAM H1 plus 21 gave your 9 kb band, which is this one, and 6 kb band. So if you divide this 15 kb plasmid, now you can see that it's like 6, 6 and 3. So which is going to give you like 15 kilobands, 15 kilobands. And their question is BAM H1 and Eco R1. So yeah. So we don't even need to take the next not one Nico R1. So automatically you can see here that if we cut BAM H1, so out of this 9, it's going to give you 6 and this 6 is going to give you 9. So automatically you can see that between the BAM H1 and Eco R1, the band size is going to give you 3 kilobase. So if you cut the plasmid with BAM H1 and Eco R1, you would eventually get your 3 kilobases and the rest of the 15 kilobases, if you cut out 3 kilobases, it will be 12 kilobases. So the correct answer will be option B, which is 3 kb and 12 kb. So do you get how this answer is solved?
Sir, can you go through it once again?
Sure. So yeah, basically this whole uh, yeah, basically this whole plasmid size is 15 kbs, 15 kilobases, and out of that they have given a couple of restriction enzymes, not one eco R1, BAM H1, and STU1. But out of that, what concerns our question, main question is BAM H1 and eco R1. So we have to see in which cases BAM H1, eco R1 are having a very common to go around this question, to solve this question very easily. We have to see which restriction enzymes BAM H1 and eco R1 are being cut along with. So if we look from the last line, it says that eco R1 and STU1 give uh, two different bands of 6 kb and 9 kb. So From this itself, we can see that like two enzymes are cutting the plasmid to give you two bands. So this automatically tells you the fact that each restriction enzyme has only one restriction site. This I think this I pretty much explained this in last week's TA session as well. The number of bands that you are getting directly correlates to the number of restriction sites that are obtained as well. So if two enzymes give you two bands, that automatically means that there are only two restriction sites, and like uh, just as a rule of thumb, those uh, the problem can be further complicated as well. But now, just for simplistic reasons, we'll consider that we'll assume that each restriction enzyme has one restriction site. If the problem doesn't work by having this assumption, then we'll try a different assumption saying that either of the enzymes will have two. So it's just basically a trial and error method. So for now, we'll assume that both the enzymes EcoR1 as well as 2,1 will have 1 restriction site each. So what we are saying here, EcoR1 plus 2,1 gives you 6 kilobases and 9 kilobases. So here we have already noted it down. The smaller fragment is going to give you 6 kilobases, and the larger fragment is going to give you 9 kilobases from EcoR1 to 2,1 the other way around. Let's look at the second question in this case. Sorry, second statement in this case. So BAM H1 plus 2,1 gave 9 kilobases and 6 kilobases. This is quite the opposite. It's not giving you the same. So giving you 6 kilobases and 9 kilobases is kind of different from giving you 9 kilobases and 6 kilobases. And here we don't have eco R1, we have BAM H1. So let's just arbitrarily assume BAM H1 is somewhere around here. Yeah. If you keep it like you can also keep it between eco R1 and stu1, but if you try to work it out, you won't be able to get the 6 kilobases and 9 kilobases because the gap will be so small, and mathematically it will not be possible for you to have 6 kilobases cut here. So, and even if it's cut here, it's going to give you like it's going to give you 3 kilobases. So for actually we can also solve it this way as well. So let this be also your BAM H1. So BAM H1 can either be between E and eco 1 and stu1 on this side or on this side. But in either cases, if it cuts, it's going to give you so the BAM H1 and stu1 is going to give you 9 kilobases and 6 kilobases. So since this is already like yeah, one second. Yeah, this is actually yeah, this is not going to give you the correct mathematical this thing because uh, if this is between eco and stone, which is already 6 kilobases, BAM H1 being here will not give you 6 kilobases again. So it must be somewhere around on the other side. So this will make more sense here. And if it cuts here, if BAM H1 and stu1 cut here, that is going to give you 9 kilobases and 6 kilobases. So if you assume this 6 and then some extra portion to be 9, this will automatically become 6. So you just need to find out how much this uh, x needs to be. So if you have 6 here, and total is 15, it will automatically give you x equal to 3. So the distance between your BAM H1 site and your EcoR1 site will be 3 kilobases. The question is, they are asking how when BAM H1 and EcoR1 cut the plasmid, what will be your following fragments? So automatically when BAM H1 and EcoR1 cut, it's going to be 3, but it's also going to give you this large fragment which is 15 minus 3, which is 12 kilobases.
Yes, sir, got it, sir. Now.
Cool. I'll once again, I like in next week I'll try to explain this more properly with a proper plasmid map. But for now, this is the.
Okay, sir, I'll leave then. If there is
Yes.
nothing more to discuss, then I'll leave.
Yes, yes, sure, sure, sure. But the room will be open till 4:30 or 4:40. If you or any of your friends want to discuss any doubts, you can just join the room and discuss it till then.
Sure, sir. Thank you, sir.
Yes, 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 sure. Okay, so if there are no other doubts for this week, I'll close the session for this right now. I'll meet you guys for week 4 session on 15th February from 3 p.m. to 5 p.m. Thank you.